Welcome to our Oxford Brain Diagnostics guest speaker series once again. By way of introduction to us, Oxford Brain Diagnostics, we are an Oxford University spin-out with a method for analysing brain MRI based on cellular structures, and we're working towards a clinical tool for early detection of Alzheimer's disease and other neurodegenerative diseases, which is to be used by clinical services and by drug developers. So I'm very pleased today to be able to introduce Dr. Jeffrey Cummings as our guest speaker. He's one of the most cited researchers in Alzheimer's disease and an advisor to many major companies, far bigger than ourselves. So we're honored to have you talking to us, Jeff. Jeff studied at the University of Wyoming and University of Washington, Seattle. He completed his neurology residency at Boston University and spent some time here, I believe, in the UK at London Queen Square, the National Hospital for Nervous Diseases. Before returning to the States and following some time at UCLA, I think. Jeff is now director of the Chambers-Grundy Center for Transformative Neuroscience at the University of Nevada, Las Vegas. I think he has many other accolades to his name as well. But Jeff, your annual publication on the AD drug development pipeline is familiar to many, and I believe you're going to speak to us about that today. So I'll hand over to you. Thank you.
Oh, thank you, Stephen, and thank you all for coming on. I look forward to a vigorous exchange of ideas. This is such an amazing time in our field that it's really just amazing. And I thought I would just jump onto this opportunity to present some of the things that we do. So I consult for a lot of companies, so I have a good sense of what's happening in the field and what drugs are moving along, and that has led us to the pipeline work. So I'll start with the pipeline overview, talk a little bit about the categories of drugs. It's pretty interesting to look at the pipeline and see what's evolving. I'll emphasize, of course, the monoclonals, just a couple slides on that because that's where the action is right now. And then Gad mentioned that he had seen the article on biomarkers and our thinking about clinical meaningfulness. And I think that is really relevant to what you do. So I thought I would also talk a little bit about that. So here's the central figure of our annual publication. We've been doing this since 2016. And it's become a passion of mine, and it is funded by the NIA now. So we have a 5-year grant to continue and to expand this work. The way the figure works is that the central circle are all the compounds in phase 3. So they are in the most advanced trials. The second circle is phase 2. That's always very well populated because there's a lot of drugs that don't make it to phase 3. And then all of your repurposed compounds are coming in at phase 2, so they do not appear in phase 1. And then the outer circle are the phase 1 compounds that we can identify. We use clinicaltrials.gov as the source of this information, and you must register all trials done in the United States on clinicaltrials.gov. Many phase 1 trials are done ex-US. So the result is not all of them are registered on clinicaltrials.gov, and we know that our phase 1 population is not fully representative of the phase 1 activity in the world. So we're working on that, just like everything else as evolves, and we're letting this evolve. The colors indicate the CADRO classification. CADRO is the Common Alzheimer's Disease Research Ontology. And it comes from the NIA and the Alzheimer's Association and another larger group of collaborators who identified a series of processes in Alzheimer's disease. And it's a fairly comprehensive list of processes. So those processes are targets for therapeutic intervention. So we use the CADRO to identify the target for the drug. So an anti-antibody, an anti-amyloid antibody would be listed as an amyloid drug, of course, and so on for each of the drug categories. That's not exactly as specific as we want for mechanism of action, but it is a preliminary classification of at least the target that the drug is involved with. So the color classification there is CADRO, and I'll take you through a little bit more of that in a moment. And then as you see in the lower left, the shape of the icon tells you whether this is a trial of preclinical disease, which would be people who are cognitively normal but have amyloid in the brain by either CSF analysis or more likely amyloid PET, or do they have prodromal AD, which would be MCI plus a positive PET, or mild, moderate, or severe Alzheimer's disease. So you can tell the population through the shape of the icon. As you see, there are 143 agents in 172 trials. Some agents are in 2 or even more trials. There are some interesting patterns that emerge. You see that there are 31 agents in phase 3, which is not a lot. And of them, 21 are disease-modifying therapies. So you see the tremendous predominance of the disease-modifying philosophy, conceptual framework that we are working in now. In phase 2, there are 82 agents, of which 71 are disease-modifying agents. And you see there's quite a few biologics in each of those. Biologics are mainly the intravenously administered monoclonal antibodies. In phase 1, we've been able to identify 30 agents. So altogether, 83% of the pipeline is disease-modifying therapies. Only 9.8% are cognitive enhancers. And if you look at the orange slice of the pie on the left, that's the cognitive enhancers. And you see that's really unexciting, right? There's caffeine over there. There's the Donepezil, which we already have. It's in another phase 3 trial. There's an octahydro aminotacrin, and we've had tacrin for a long time. It's gone off of the market now. So there's really a huge open space for cognitive enhancers. I should have pointed out, by the way, that the green slice of the pie are all the biologics. The purple slice of the pie on the right are all the small molecule disease-modifying agents, and the blue slice of the pie at the bottom are the neuropsychiatric symptoms. So only 6.9% of the drugs in the pipeline are devoted to neuropsychiatric symptoms like agitation, but they have a disproportionate chance of succeeding because, let's take brexpiprazole, for example. Brex is approved for schizophrenia, it's approved for bipolar illness. Now it's being tried for agitation and Alzheimer's disease. Well, we know a lot about that drug. And the likelihood that we can make it succeed in a trial is pretty high. It's not by no means a given, but it's pretty high. Much higher than taking one of the disease-modifying small molecules and trying to make it work for the first time in any way. It is also interesting that 37% of the agents in the pipeline are repurposed. So that means they're approved for some other indication. Let's just look at, if you look in the purple slice of the pie, phase 3, you see metformin there, for example, approved for diabetes. You see nilotinib, that's approved for cancer. You see semaglutide, that's approved for diabetes, and so on. So there are many repurposed agents in the pipeline, and I'll talk maybe a little bit more about that later. So this is how it works. This is the main figure. People take a screenshot of this and show it in their lectures, and I love that. So please do it, and we love it that this gets used. It's alternately called either the bullseye or Jeff's wheel of fortune, whichever you like. So feel free to use it. And by the way, I get a lot of correspondence about it to make sure that, someone says, Jeff, you didn't get my agent in, or you have the mechanism not quite right. And so it's almost a community project now, and we enjoy that part of it as well. So now I'm showing you another way of looking at the pipeline, and this is using the Common Alzheimer's Disease Research Ontology, or CADRO. I'll start at the bottom there. You see how this is divided into the targets of the disease: amyloid, tau, inflammation, synaptic plasticity, metabolism, proteostasis, vascular, oxidative stress, epigenetics, neurogenesis, transmitters. And you see right away where the action in the pipeline is. It's in immunity and inflammation and synaptic plasticity. Amyloid is still a popular target, but you can see that there's a lot of interest in non-amyloid and non-tau targets. And we don't have too much going on in tau. There's 9 agents in phase 2. That's encouraging. We'll see how that goes. One of the things I didn't mention, by the way, that is kind of an orienting factor is you have to choose, when you're writing a paper like this, you have to choose one day in which everything you say is true. So everything that is I present was true on January 25th of this year. So if an agent came into the pipeline on January 26th, it isn't here. And that frustrates investigators, who say, where's my compound? Where's my compound? So we adhere strictly to an index date. And of course, that makes sense of all of the information. But you'll see that there are some compounds that might have dropped out by now, and some compounds that might not be here because of the index date phenomenon. But I think you'll find this interesting. The metabolism and bioenergetics classification, by the way, that is all the insulin drugs. There's a lot of enthusiasm for them now. 2 huge trials with semaglutide, for example. We're seeing more of the epigenetics coming in as well. Just to show you a little bit about how some of these very well represented areas work. On the left, I'm showing you a cartoon of inflammatory processes in the Alzheimer's disease brain. So the resting glia is transformed to the activated microglia, and they release many pro-inflammatory and anti-inflammatory cytokines. And it is the balance between those and the engulfing activity of the microglia of the amyloid that determines the ultimate inflammatory state of the brain. And there are very many contributors, including amyloid itself in the oligomeric form. Neuronal death is a stimulant to inflammation. There is peripheral inflammation that can enter the brain through the blood-brain barrier. And we think that's a major part of the inflammation that is seen in both diabetes and obesity, and why those are risk factors for Alzheimer's disease, for example. But I want to call your attention to the agents on the right. These are all of the agents that are in the pipeline for the treatment of the inflammatory process of the brain. But note that there are almost no categories in which there are 2 drugs. So you start at the top, there's a TREM2 antibody. There's an immunomodulator. There's a non-steroidal anti-inflammatory. There's a tyrosine kinase inhibitor. There's a T-cell regulator. There's a cytokine antagonist. So we're really going to learn a lot about inflammation because there's so little overlap in the anti-inflammatory drug mechanisms that we're seeing. I think some of them are, of course, in more strongly developed development programs than others. So you have to take that into account as well when you see the success of a drug. But I'm interested in trying to understand better the many mechanisms and how to conceptualize that. One way that we're currently thinking about doing is we have maps of networks of disease activity in Alzheimer's disease. We also have maps of how each of these drugs affects a system of genetic expression. We now have proximity scores for how close those 2 things overlap with each other. And we're going to start looking at this whole list of inflammatory drugs to see, are there some standout compounds where the effect of the drug would predictably have a greater effect on the inflammatory processes. So just to give you a little sense of where we're going and what we see as exciting here. And then the other big category that's in the pipeline outside of amyloid and tau are the synaptic plasticity idea. Synaptic plasticity is affected by oligomers. It's also affected by inflammation. Of course, neuronal cell death limits the synaptic integrity. And for me, I'm excited about this because the synapse is the building block of the circuit, and the circuit is the basis for human cognition and emotion. It is not something as limited as a synapse. It is the integration of the neuronal activity that we're trying to restore or to slow the decline in our Alzheimer's disease therapeutics. And it opens up, of course, a new set of measurement opportunities like functional MRI, for example, and EEG as means of assessing circuit disruption, and can that be restored through these synaptic drugs. Again, I have the list of agents on the right, and again, just look at how many sub-mechanisms are involved. GABA B modulators, sigma-1 agonist, PDE4 inhibitors, there's a plasma transfusion approach. Levetiracetam is actually, I think there are now 6 trials on levetiracetam, which is an SV2A modulator. And that's particularly interesting because we have the new SV2A PET scan. And so it allows us more insight into the synapse from a PET perspective than we have had before. But again, you see the diversity of approaches to a single kind of pathology in the brain is an opportunity to learn about which of these might be most important or which of these might be most manipulable. So let's go now to the monoclonals, and we can come back and talk about any drug you want to talk about, but I just chose a few things I thought you might be interested in. So again, on the left is the process. You have the amyloid precursor protein, 770 amino acids long. The portion that is 42 amino acids long and is in the membrane and is cleaved first by beta secretase and then by gamma secretase, frees up this 42 amino acid linked monomer. When it's 42 amino acids, it is particularly prone to aggregation, so it forms the A-beta oligomers, and that goes on to fibrilize and form the A-beta plaque. So the plaque is known as the insoluble form of A-beta, and all of the predecessor forms are the soluble forms of A-beta. You know that with amyloid PET, we are removing only the plaque. So it doesn't give us very much insight into the rest of this. In the spinal fluid, we are measuring only the monomer. So we can see the two ends of this cycle. We can see the monomer in the CSF. We can see the plaque on the amyloid PET, but we can't see anything in between. And those might be the most important species. It's a conundrum right now for the field. So where are we with the monoclonals? Aducanumab, as you know, has been approved in the United States. A lot of controversy there. It clearly lowers A-beta plaques in a dramatic way. And in all of the testing that we can see, it also affects oligomeric A-beta. And I have just said that we have no measure of oligomeric A-beta. So that is based on animal model work and cell models and that sort of thing. Donanemab is in phase 3. It clearly also has this great A-beta plaque lowering effect. It focuses specifically on pyroglutamate A-beta. The interesting thing about that is pyroglutamate A-beta is found only in the plaque. This probably means that plaque reduction itself is important. It is a drug that we would predict is not affecting the oligomers. So again, it gives us an opportunity to learn by contrasting the effects of drugs that have different mechanisms. Also in phase 3 is lecanemab. It lowers A-beta plaques, and it was specifically raised to target the protofibrillar A-beta. It will read out this year. The Clarity trial will come to an end before December, and we will know by the end of this year, 2022, whether lecanemab is effective or not. It's really going to be exciting. The same is true of gantenerumab. It focuses on the plaque. It also affects oligomeric A beta. It's in phase 3, and the graduate programs are going to read out this year. So in the second half of this year, we are going to learn an enormous amount about Alzheimer's disease therapeutics. Now, there are two drugs that you hear much less about, but they're also going to read out soon. One is crenezumab, which affects both oligomeric and monomeric A beta, and is a phase 3 trial of autosomal dominant Alzheimer's disease in a large Colombian family. And all of the members of the trial have confirmed mutation carriers. Well, that's not quite right. Because they didn't want to reveal the mutation status, they included a few non-mutation bearing individuals in the trial and made sure that they got placebo, just so that no patient actually knows whether or not they might be that person who is a non-mutation carrier. So that's going to read out this year. And solanezumab, which was focused really on the peripheral A beta monomer, and you see in the figure on the left that you can have peripheral monomers that enter the brain. And the idea was you create a peripheral sink by binding the peripheral monomer, and you can pull the central monomers out. That did not happen in 3 well-done phase 3 trials by Lilly, but it is still in a phase 3 prevention trial where people were normal, cognitively normal, with positive amyloid PET at the beginning of the trial. And that trial will read out, I think, either this year or next year. So there are a lot of monoclonals in play right now. There are more coming. We're going to learn a lot about this. Aducanumab had a very difficult development program. I'll go through that in just a moment. But Donanemab and lecanemab and gantenerumab that will also be available this year, I think will have much less controversial, much more regular clinical data that we can have a different kind of confidence in. The Oxford Brain Diagnostics is a biomarker group, so I wanted to emphasize a little bit about biomarkers in this discussion. So here's the way the FDA approaches it. Biomarkers are in one of 7 recognized contexts of use. You guys work in this area, so you probably know this, but not everybody does. So a biomarker must be in one of these areas. And a biomarker would always be defined as having a context of use. So as you see there, it's either a risk, a diagnostic, a monitoring, a pharmacodynamic, a predictive, a prognostic, or a safety biomarker. And one biomarker might have several contexts of use. For example, you could use amyloid PET for diagnosis, and you could use it as a pharmacodynamic biomarker for amyloid reduction. So that's common. But both of them would have to be declared, and both of them would have to be qualified, that both context of use would have to be qualified. And there's a qualification process through the FDA that allows a biomarker to become qualified. If it's a qualified biomarker, then the readout of the trial can include that biomarker information, and it wouldn't necessarily be exploratory. So these are nuances that are very important in terms of what can be claimed that a biomarker is reporting. I put on the right there some of the examples of the biomarkers that we currently have in AD. So of course, ApoE is a risk. ApoE4 is a risk gene, so we can measure that. Amyloid PET or CSF-A beta for diagnosis. Amyloid PET, tau PET, plasma p-tau are all monitoring opportunities. The ATN framework I would regard as target engagement biomarkers. The difference between predictive and prognostic is important. A predictive biomarker tells you that there is one group of patients who will do better or who will do worse when given a drug. So prediction is related to the drug. Prognostic biomarker tells you what's going to happen to the patient. So a patient who has elevated amyloid PET has elevated amyloid PET and elevated p-tau217, for example, is going to progress and probably progress pretty soon. A safety biomarker, of course, we use MRI for ARIA, but we also use liver functions and EKGs, and those have become so common we don't even think about them as biomarkers anymore, but they are. Just a few points that I made on the left there. The role in the trial depends on the defined context of use. The MRI, such as Oxford Brain Diagnostic, could have several roles. So you could imagine a monitoring role, perhaps a pharmacodynamic role, maybe a prognostic role, maybe a diagnostic role, which I didn't include there. So those are all context of use in which I could imagine your technology. So let's go on here. Let's see. I just wanted to give you an example of how these biomarkers are now being used in trials. And I'll just use the Trailblazer, the Donanemab study as an example. So tau PET was used to define the population. A patient had to be tau positive, but could not have overabundant tau. So it was sort of the Goldilocks solution between too little and too much. That was the population that was brought in. When you define the population that way, almost everyone is amyloid PET. There was only a 0.9% disagreement between amyloid and tau PET in this study. So that's an example of a diagnostic biomarker. You see that below the impact of removing the A beta on the tau was also measured, so tau PET became a pharmacodynamic biomarker. In the upper right, you see the p-tau217. So as you pull the amyloid out of the brain with donanemab, you can see also that the p-tau217 is going down. So this is a downstream effect, and that is a monitoring biomarker that you see was collected every 3 months to determine what was happening over time. So that's an example of a monitoring biomarker. And then you see the reduction in amyloid plaque in the lower right. You see there's already a 70% reduction at 6 months. I mean, these antibodies are just so effective. We've never seen anything like this. This is just amazing that you can do this much to the disease process. This was an interesting trial in that if the amyloid PET was shown to have had this kind of dramatic response, some of the patients were then re-randomized to either drug or placebo, so that you could watch how quickly amyloid reaccumulated in the group that was re-randomized to placebo. Of course, it stayed down throughout the entire trial, raising provocative new questions. Now, Ged had asked about the measures of meaningfulness, and I'll get to why this is important. So in the upper part of this, you see measures of clinical meaningfulness that have been used in the symptomatic stages of the disease. So this would be mild cognitive impairment and mild, moderate, and severe AD. In MCI, you can use a single composite measure. That would be, for example, the CDR sum of boxes. In mild, moderate, and severe disease, the FDA requires dual outcomes. So a common one in a common situation in mild to moderate disease would be the ADAS-Cog plus either the ADCS-ADL or the CGIC. And the idea that the FDA presents is the ADAS-Cog measures the core features, which must be done, and the impact that is global or on function is a measure of clinical meaningfulness. So that's how that came along, and that's also why a composite measure like the CDR sum of boxes, which 3 of the components are functional, is also regarded as clinically meaningful. Patient-reported outcomes are just a great source of interest to the FDA right now. We don't really have them in Alzheimer's disease. What we're getting is proxy PROs from the caregivers, and there still needs to be a lot of work. But of course, what's important to the patient is by definition clinically meaningful. You can measure effect sizes. This is often a good thing to do in smaller trials, and you can have small, medium, and larger effect sizes, and that gives you some idea of meaningfulness. You can have relative risk or an odds ratio of improvement or an odds ratio of decline, and that can be seen as clinically meaningful. Payers like these number needed to treat, the NNT, or the number needed to harm, the NNH, and that determines whether they will be comfortable reimbursing a drug. So that's their measure of clinical meaningfulness. Another measure that I like a lot, but not everybody does, is time to event. And a common one is to have a population that is either all MCI or half MCI and half dementia, and you measure how much longer it takes for the treated population to progress from MCI to dementia compared to the placebo group. So, and that's meaningful, right? The delay of onset of the advanced phases of Alzheimer's disease is a meaningful outcome, and people relate to that as such. And then there's a measure called the MCID, the minimally clinically important difference, and that is also seen as a measure of meaningfulness. Now, our challenge was, and we put this into the publication, we struggled with this a lot, is how do you decide what is meaningful if the population is in the preclinical state, right? These are normal people. How can you make normal people better? So the idea of clinical meaningfulness starts to be eroded. And we are trying to move the field towards measures of meaningful benefit rather than clinical meaningfulness, because I think clinical meaningfulness implies that you're doing something that the patient can perceive or that you can perceive. So we have proposed two measures of meaningful benefit in asymptomatic populations. One is cumulative benefit, and we know that in disease-modifying therapy, there's an increasing drug-placebo difference over time. So if you have a certain slowing at 6 months, then you'll have greater slowing and proportionately greater at 12 months, 18 months, and the longer you stay on the drug. So we've called that cumulative benefit. And the other is predictive benefit. Remember, I just told you the difference between predictive biomarkers and prognostic biomarkers. So if you had a predictive biomarker that would tell you that the change in the biomarker will predict a clinical benefit, then that would be a meaningful measure of the activity of the drug in an asymptomatic population. So the biomarker anticipates the clinical change. So those are some new ideas that we've just put into the literature, and we're happy with these. So I'll just summarize what I think is important and what I've been thinking about, and I'm really looking forward to the discussion. There are many mechanisms represented in the pipeline. I've emphasized inflammation and synaptic plasticity, but you see that virtually every CADRO classification is represented. The biomarkers are enormously helpful. I just showed you Trailblazer as one example. There are many examples. I think the idea of doing drug development without biomarkers is almost unthinkable at this point. But we need a lot more. You think of the thousands of cellular processes that are involved in Alzheimer's disease and how few biomarkers we have compared to that. So we need a lot of work. Most of the drugs are in the pipeline are disease-modifying therapies. You saw that 83%. There are very few cognitive enhancers and drugs addressing neuropsychiatric symptoms. There's a lot to do there. Remember that there were 143 drugs in the pipeline. If this was a cancer talk, there'd be 3,000 drugs in the pipeline. So you see how far we have to go and how much work there is to do, but we're making progress. And aducanumab is the first disease-modifying therapy for AD that's been approved by the FDA, very controversial, as you know. But I think we're going to have a lot of clarity towards the end of this year when the other anti-amyloid monoclonals are read out, and we'll really have a lot of information on their clinical benefit and on their effect on amyloid. We can already see a lot of the effect on amyloid. So this is my team. I work with although I'm in Las Vegas, I work mostly in terms of our pipeline work now with the Genomic Medicine Institute at Cleveland Clinic in Cleveland, but I have a whole variety of great collaborators on all aspects of the work we do. So thank you, and I really look forward to having some questions and answers here.
Thank you very much, Jeff. For a wonderful, comprehensive review, actually, and also very thoughtful. Thank you for thinking a little bit about how Oxford Brain Diagnostics tools might fit into that. I mean, not only obviously your direct references are useful and food for thought for us, but I was thinking also about in terms of groupings of drugs, those that are targeting the synapse. And you mentioned effectively the kind of integrity of the circuits. I think that's quite interesting because although you referred to functional types of assessment, fMRI and EEG, in many ways, I think we consider that we're assessing neurodegeneration and we're looking for the structural corresponding features. So because we're looking at microcircuits, you know, our target in the original design of our method was around the mini column structure, which is the ubiquitous sort of circuit feature of the cortical layers. So I think that's very interesting that we might particularly apply ourselves to that area. But of course, a marker of N in the ATN framework is very much where we see ourselves sitting. So in multiple ways, I think we are relevant to this picture. But that's really just a comment, a bit of positioning, if you like. But I'm not anticipating too much about which questions will come because the whole team will, I'm sure, have many questions. Who would like to ask?
I'll just get back to that, Stephen.
Yeah, sure.
I wondered about that when I looked at the website, whether your measures would tell us something about circuitry, because you probably know that it's been very difficult to do fMRI at the multi-site level and have it be really solidly reproducible.
Yeah.
So if we had a structural measure that we could use instead of fMRI, it would have a great advantage.
Yeah.
So I would encourage you to keep working at that, because we would love to have measures that, you can put, I don't know, 140 sites in some of these trials, maybe even 200 sites in some trials. Well, that's ridiculous in trying to do fMRI. So an alternate measure would really be valuable.
Yeah, thanks. Absolutely. That's what we hope. That's part of the challenge we hope we can address. Yeah. Right. Who would like to ask a question? Terry's up first up.
Hi, Jeff. That's very interesting. Before we jump into the technical details, I was just wondering if you could go back, 5-10 years, and you could give some advice to the research community, knowing what you do now. What would you tell them?
I would tell them that the trials they are doing cannot give us an answer because there was no biomarker readout. There was no definition of who was in the trial. They were clinically diagnosed patients. We know now that at least 20% of them don't have Alzheimer's disease. And the earlier you go, the fewer of them have Alzheimer's disease. To me, as I look back now, Terry, I think it's a miracle that the cholinesterase inhibitors worked as well as they did and had a measurable benefit. Because the trials were just so poor compared to what we would consider a well-done trial now. The answer is in biomarkers, because that both allows you a less heterogeneous population that actually has a disease of interest and allows you to measure the impact on the biology. If you have a well-diagnosed population and you put your drug in at the maximum dose and nothing happens to a biomarker, I think that drug program should end, right? Because if nothing happens to the biology, I don't see how we can predict that something is going to happen to the clinical end of things. Now, you can have something happen to the biology and it's not enough to change the clinical picture in a measurable way. That's also a possible outcome. But if you don't change the biology at all, I just think we should stop. Use those resources to do something else, to develop another drug. So that's how I would think about it. But of course, we just have technology and tools now that allow us to take that position that weren't present then. So we were all doing the best we could at the time. We thought we were doing great science, right? And now we can see in retrospect the many faults that were present.
Thanks.
Great, thanks. Jed, there's lots of questions. Jed, I think you were just first.
Actually, something of a quick follow-up to that, if I may, on the question of kind of moving the biology. I saw earlier this week the paper on the tau reel trial with a slightly confusing picture with changes in CSF p-tau, but not in tau PET. How would you interpret that in terms of, is that a positive or a negative signal overall in terms of biology? Could it simply be that p-tau is, there's evidence, I think, that it's changing earlier than tau PET, and that tau PET's more of a kind of cumulative effect. So could it just be about the dynamics? Could it just be that the trial was too short? I'd be interested to hear your thoughts on that.
I would say yes and yes. We do know that they're providing different kinds of information, aren't they? Because p-tau is changing really early in the disease, and you don't see any change in tau PET. And then the tau PET starts to change at about the same time patients get symptomatic, more or less. And you see that medial temporal signal first, and then it spreads out to the rest of the telencephalon. So we know that the time frame is different. So it is possible that this is just a dynamic. It's possible that one is simply easier to move than the other, and that would again relate to the dynamics. And then the sample size and the duration of the trial may have been inadequate. One of the things that surprised me about the aducanumab trial, and now I guess also donanemab, is that you do change tau PET. I can really see changing p-tau, but I'm surprised that removing amyloid has an effect on existing neurofibrillary tangles. So that is a more dynamic relationship than we appreciated. The great thing about data is that they change our minds if we're open to it. So I think we're learning about this dynamics of different measures of tau that we haven't seen before. We now have 3 p-taus, of course, 181, 217, and 231. Both 181 and 217 seem to change pretty early, and then 231 seems to be telling us something about the, sort of a later form of the disease, maybe a little bit before the tau PET, but after 181 and 217. And there's a surprisingly close relationship between plaque reduction and 181 and 217. It's as if that epitope is associated with the plaque, the neurite in the plaque, because they're so tightly linked. All of which is going to be highly informative, I think. In the U.S., we have 2 approved blood tests for A beta 42 to 40. And I think we will very soon have an approved p-tau test. Now, when I say approved, they're approved to be done in one laboratory. They're not done at the kit level where you can buy a kit and have it done. So there are some nuances there about how approved they are, how rigorously approved they are. But they're on the market, you can buy them, and you can use them every day.
Thank you.
I think, excusing background noise, I think Omar was just next, and then Dimitra. So Omar, you go ahead.
Thanks, Stephen. Dr. Cummings, can you hear me? I've just had to switch to my phone.
Yes.
Oh, fantastic. Wonderful. I just wanted to pick up on, I think it's a lead into Terry's question, and it's wonderful that you mentioned some of the things you did with the beauty of hindsight. You also co-authored a paper on the rights of precision drug development, where it looks at the 5 rights for AD clinical trials. Have you had any thoughts on how best we could integrate that more consistently in your annual review of the AD pipeline? Because I just feel like if we get into 5 years now, possibly even to 10 years, we shouldn't lose sight of those 5 rights when we're running these clinical trials. And I just would like to see an annual update of it.
Yes, thank you for that. I love that rights paper. We had so much fun writing it and thinking through it. And of course, it was all stimulated by the early AstraZeneca work. You know, they came up with five rights, but those were mainly based on cancer drugs. But some of them clearly applied to other therapeutic areas. So that's how we wound up writing the 5 rights paper. And here's why I don't do that, Omar. These are all people that I love who are doing these drugs. And if I actually went through the 5 rights and showed how bad their trials are and published that, I would lose all my friends. There's a surprising number of trials that are still being done without proper diagnosis, without proper outcomes, without proper power. You know, they would have maybe 1 or 2 of the 5 rights, and those trials are almost certain to fail. I've thought about doing something about that, but I am really reluctant to be too critical. It's not in my nature to be as combative of that. I think we could take a slice of the pie, maybe, and look, for example, at all of the synaptic drugs and do, you know, a more limited analysis and say, well, here's how synaptic trials could be improved, and here's where it looks like they have a consistent weakness, for example. So I think, and I've been thinking about doing exactly that. It needs to be done. It needs to be pointed out. And here's what's frustrating to me. The NIH is funding trials, sometimes with lots of money that would not meet the 5 rights. I think that the review process should be more scientifically rigorous than it is in terms of drug development if you're going to fund drug development. And we're just not seeing that at this point.
Thank you.
Yeah, I think those are very powerful points, and we often talk about in the company the sort of concern about the sense of lots of resources thrown at the whole field, and of course, as you've said, things have moved on a long way, but still there's a sense of needing that rigor and agreed standards and so forth that's very difficult to, which I suppose, to achieve consensus, of course, about. But yeah, anyway, so Dimitra, I think you were going to ask a question, is that right?
Yes, thank you for your talk. MRI and PET are commonly used in AD trials, but often with simple measures like whole brain SUVR or hippocampal volume. Do you think trials could make more use of the information on spatial patterns? That is a strength of imaging over fluid biomarkers.
Yes, absolutely. I think there's so much more structural information than we're making use of at this point. And I think we could get to a point where we wouldn't need amyloid PET. That would be sort of at the extreme end of what could be done. But I'd love to see you guys working on that. And I'd love to see, more use of these markers. So, I practiced for 10 years in Cleveland Clinic and led the Lou Ruvo Center for Brain Health here. We had a simple program called NeuroQuant. It's commercially available that gave us a hippocampal measure, and I found that very useful. And that's like first generation compared to what you are capable of. I think that there is much more structural information that's available that we're not making proper use of, and there's part of it. It'll be good to get out of the monoclonals for that particular issue, because when you move the amyloid, you routinely increase the atrophy. That's been seen in almost every trial. Well, then what do you do with that? How do you interpret that when we think that, of course, atrophy is not what we want, right? We're trying to have less atrophy, not more atrophy. But the dynamic of removing the amyloid is you increase the ventricular size and you decrease the total volume. But that it hasn't been exclusively seen in the monoclonals, but it's much more likely in the monoclonals. So I think applying this technology outside of the biologics will probably be more rewarding.
Thank you.
Yeah, that's a very interesting thought. Certainly we take the view that volume is not the whole picture. So it could be an advantage to have less of something as long as the quality is good. So that's one of the reasons we look. We want to achieve a measurement which is about cellular scale change, because that might reveal things that are not seen at that volumetric level. So.
Well, the other thing that I think is really interesting is, if you look at our prodromal trial of varabacestat, those patients have atrophy induced right away. At 3 months, they have more atrophy. Mm-hmm. And then they parallel in decline the atrophy of the placebo group thereafter. Well, that's just, that cries out for a structural understanding. So I would love to, see some more high-powered kinds of analyses and technology applied to issues like that, where we really need to understand, why did a BACE inhibitor make people worse? There could be a biological contribution to that as well as a clinical one.
More questions from the team. Jamie, just ahead of Michaela, yeah.
Hello, thank you. That was a really interesting talk. My question kind of refers to how you do feel that aducanumab might change the clinical trial landscape, and particularly in cases where patients who are enrolled in trials might be being treated with aducanumab, and would they be excluded or included, but specifically included for almost as a control group, or how do you think that might balance out?
Yeah, really great questions. The first thing I would conclude, don't do a futility analysis, because that's what changed the ability to draw any conclusions really from the kind of conclusions we'd like to draw from the aducanumab trials. But more specifically, what about trial participation? There are going to be so few patients on aducanumab. CMS isn't going to pay for it. It's probably running about $20,000 a year, which is not actually too bad for if you compare it to cancer drugs, for example. But it's enough that it's going to keep people off of it. It probably has a higher rate of ARIA than other monoclonals. We don't know that for sure yet, and it's hard to compare across trials because they all have these little subtle differences that might play themselves out in the ARIA frequency. But it looks like it probably has more ARIA. So I think it's going to be such a small percentage of patients in which this question comes up that I don't think it is going to pose a challenge for us in terms of drug development or clinical trials. I'm so regretful of the CMS decision here, because I think the data were enough that I wanted my patients to be able to, and my caregivers to have a voice in that decision to decide whether the risk of harm was worth the apparent possibility of benefit. I wanted to be able to present that to my patient, and I really can't when CMS won't pay for it. That was the scientific community was really very fractured by this. And I was, on one side, but that's where I came down on this, and that's why I think we're in a temporary period where there's going to be a lot of clarity. Even at the end of this year, we're going to have a lot of clarity. I think we're just in a transit period. We're growing, learning new things, and there's a lot of irregularities about it.
Great, thank you.
Yeah, I guess from the end of this year, we might have some other drugs that will have a larger, wider uptake. And then I suppose in a way, Jamie's question may apply in a sense to those as one considers, in comparison with some of the other areas of other targets that we've talked about.
Yeah, I know we're coming to the end of the hour here. The CMS said that if they got clinical data, what they would require of the new antibodies is a registry Well, that registry could be very easy or it could be very difficult. And what the registry looks like is going to have a profound effect on how many patients will go on to the drug, what effect it might have on trials, how it plays itself out in the field. If it's a kind of a sign up, it'll be easy. If there's a lot of data involved and a lot of monitoring involved, it will very much limit the use of the drug. So we're in a period where we don't know what CMS's registry requirements will look like, and I'm sure that both Roche and ASI are talking with CMS now to try to understand that.
Do you have 2 more minutes for Michaela's question, Jeff?
Yes.
Okay, dive in.
Hi, thank you very much for your talk. I was just wondering how long you think it's going to be before, or if disease-modifying drugs take over symptomatic treatment, since you mentioned that while most drugs in trials now are disease-modifying, the ones that are most likely to be approved are symptomatic treatments. So do you think there will be a point where we have more drugs that target the biology rather than the symptoms?
Yes, we're clearly moving in that direction. On the other hand, I think we'll never catch all patients early enough that they won't be symptomatic. So we'll need symptomatic therapies. And I hope that companies, pursue that more vigorously than they have, because I think we really need those. When a patient comes into me, they want to get better. They don't necessarily want to get worse more slowly, right? They want to get better. And I can convince them that getting worse more slowly is a good thing, but I would also like to give them something that made them better. So I would just love to see companies pick that up a little bit more. But there was a very special biology of the cholinergic system where there was a presynaptic degeneration and postsynaptic integrity during part of that disease. And therefore, if you add a cholinesterase inhibitor, you can push that postsynaptic neuron. And there are very few circumstances in other transmitter systems in Alzheimer's disease in which that exact biology was reproduced. And I think that's why it's so hard to push these systems. Is they're all degenerating together, both presynaptic and postsynaptic. So you don't create this therapeutic opportunity that the cholinergic system did. So it was a unique circumstance.
Yeah, thanks very much, Jeff. I mean, that reminds me, the whole precision understanding is what we really need, because that's how you sort of start to tease apart these systems. And that description you've just given for a drug action that's in many ways somewhat in the past now, is still an example of that, I think, in so many ways. So I think we're out of time. I'd like to thank you very much for joining us today. The description you gave at the start about your Wheel of Fortune and how widely is appreciated around the world, I think I'm sure you're getting fan mail from all over, as well as criticism and requests for inclusion. But I'm delighted to hear that's got funding as well to continue, and long may it continue. Thank you very much for all of that and all of the insight you've shared with us today.
I really enjoyed our discussion, and I hope your biology or biomarkers really make a lot of progress. We need them so much. So get back into the lab, get to work.
Thank you very much.
All right.
Thank you, Jeff.
Bye now.
Bye-bye.
Take care.